So I'd just like to introduce our final speaker, and we're very happy to have you, Theo, with us online. Thanks for joining us. So it's his talk is relative to this morning's section on focus on new tools for clinical research. And so he'll be focusing on nasal epithelial cells and personalized medicine. So Theo, it's over to you.
Great. Thank you so much, Natalie, and I appreciate very much the invitation. Thank you, everyone, for your patience. Can you hear me okay?
Can everyone hear? Yeah, yes.
Yep, people can hear. Good. Okay. So, yeah, as Natalie mentioned, today I was going to talk a little bit about nasal epithelial cells and how that may be relevant in the context of ANE. Um, so first, and the slides are advancing okay?
Yes. Yeah.
Great. So I was just going to, in the next 20 minutes or so, talk about the pros and cons of nasal epithelium and just even explain why we would bother sticking a brush up someone's nose in order to study A&E and talk about some of the outcomes that we can look at. So a few years back, Alex Platzo, who I know is there, had asked me to get involved with some of his amazing research. And it begged the question, why would we study the lung and nasal epithelium in this? And so, as I was thinking about this, and I know this is something you guys are aware of, when A&E was first described, I think there was this black box here, infections and the actual outcome. But at least people had linked respiratory infections as playing an important role. Over time, I think people started to refine some of this understanding, but I think there's still this black box here. So we know that infections are involved, and there's something to do with serum cytokines initially. And certainly there was this idea that cytokine storms play a role, and in particular a specific molecule called interleukin 6, which was found to be elevated in many patients. And so the theory was that maybe an infection did something, and you had more cytokines, but still a bit of a black box. I've drawn these arrows just out of my own volition. I think sometimes maybe these arrows point to each other, but for now, black box. And then in the past couple of decades, people have understood that there may be genetic variation that helps explain why some individuals are more predisposed to this. And I think that in terms of cytokine storms, people maybe have toned this down more into saying there's an inflammatory response and maybe an exaggerated inflammatory response. So when you think about these things, I think for at least for me as a respirologist and an epithelial biologist, it makes sense to study the lung because um, when you look at viral infection, especially respiratory viral infection, the main target cell is the respiratory epithelium. More recently, um, refining this, I found this review article, and I know there's many of you have looked at these data. But this was a case series, and then they supplemented their 19 cases from a group in Beijing with an additional 24 kids. And you can see that most of these cases are related to influenza virus. And for some of the other pathogens, the majority have something to do with the lung. So again, why study lung epithelium? Because it's the primary target of respiratory viruses. We know that lung epithelial cells can produce cytokines, and they do regulate inflammation. In the olden days, we used to think of the epithelium as a bit of a barrier, but now we know that the epithelium is a very, I guess I could say, intelligent cell, but a gateway cell that is very important for all the downstream stuff that happens after respiratory viruses interact with the host. So when you're studying the epithelium, I put up these two slides just to illustrate the enormous breadth of different types of cells you can study. So when I was doing some of my PhD studies, I worked with some of these are all, I mean, it just keeps going. There's lots of cell lines you can study that mimic the lung. The advantage of these cells is that they're cheap and they replicate very easily. And for a new PhD student like myself, it's very hard to kill them basically. And so you can do lots of neat experiments with them. But in our lab, in my lab, we use a primary model system. And this is just a cartoon or a schematic showing what would happen if you came to visit my lab. Well, we may have coffee or something, but also we would end up brushing your nose. Um, so we end up using primary human epithelial cells. So primary means that these are cells that are not immortalized. They come from humans and they will eventually stop dividing. Um, use a small little cytology brush, and right now our research nurse Julie would insert your nose, brush your inferior turbinate. We'd get some cells, and we would expand them in what's traditionally known as submerged culture. So that's what this little flask is. Submerged culture meaning everything is under liquid. And then somebody in my lab, either Hong or Tarini or Wendy, will expand the cells, and eventually we grow them in an insert. And you can see at the bottom corner here. I don't know if you can see my arrow, but yeah, you can. Okay. This arrow is just holding a little insert, and there's a semi-permeable membrane, which means the cells can sit on the top but still get nutrition from the media at the bottom. And this allows them to grow in an air-liquid interface. And because they can grow in an air-liquid interface, they can differentiate and they mimic the airway found in a human lung. And then this is just a picture, a histology section of the slides, and it's a cross section. The blue are the nuclei of the cells, so you can see all these cells here, and you can see the little cilia on the apical surface, on the surface facing the airway. Um, for those of you who aren't medical people, cilia are the little hairs that line our airways, and they help move the mucus around. In traditional cell culture, you won't see cilia, but in our primary cells grown at air-liquid interface, you start to see cells differentiate, and you can see all the different phenotypes or all the different changes that you see in a natural human airway. I've also put in CFTR here in green. CFTR is a protein that's found in epithelial cells, and the reason why I've shown this here is because it's the protein that's broken in cystic fibrosis. And a lot of the work that we've done has been funded by cystic fibrosis research, in part because CF is a very well-funded um, area, and it ends up allowing us to do all sorts of work, both within CF, but also building models that can be applied to other areas like ANE. Um, okay, so that's just a bigger picture, so you can see that. I also wanted to show this to you because this is also something I think, can you appreciate that on the video that the little cilia are moving here? I
Yeah,
don't know if that's,
yeah, we can.
you can see that. So that's something that just to show you that these cells are they have very motile cilia, and it's something that we can measure, and it's not something that if you're a cell biologist, you're not used to seeing your cells moving around like this. But even here, I just want to show you when we look under the microscope, the cells are there and they just move the debris around. So these are a nice model system for studying airways. Okay. So there are a few disadvantages of this model, and a reason why not everybody just decides to set up shop and make air-liquid interface cultures. One is the costs. So it'll cost in Canadian dollars a few $1,000s to go from one patient to a plate of cells for study. So that's very different from, a few $100s for a cell line. You do need some technical expertise. So this is something that took us months and months of optimizing in order to get our cells to grow. And even now, when we have new members come to the lab, it takes them a few months to get their hands wet. Is that the right word? Maybe not, but to get the expertise to work with the cells. I tell people the cells are a little bit like babies, where you have to be very gentle with them, whereas a cell line, you can be very vigorous with your pipetting and things, but with primary human cells, they tend to be a little more fragile. The other downside is there's limited expansion. And what I mean by this is when we start with a certain number of cells, we can only allow them to divide and grow to a certain point, and then they just stop. Um, Whereas with cell lines, they will expand indefinitely. So the limited expansion means when we get material, we have a few experiments we can do, and then we may have to go back and get more material. Um, so this is where the limited material comes in as well. Um, Also, by limited material, what that means is a cell line, you can go to any lab supply company and buy them. Whereas primary human cells, obviously, you need to have clinical connections and or clinical research ethics in order to be able to even obtain cells. We've learned that these cells are very difficult to modify genetically, and in the current age of science, people are very interested in changing genes or altering genes. Um, Primary human differentiated cells don't like to be genetically modified, and so it can be tricky to study the genes you want to. The other thing that's important is when I say donor heterogeneity, what I mean is that there's differences between people. When we study cell lines and we have many plates of cells, we know that genetically they're all the same and they're going to generally behave the same as long as we treat them that way. But with our cell lines, we get huge differences between individual A and individual B, and sometimes that those differences can be difficult to explain because we're assuming that we've controlled everything, but clearly there's lots of differences between the cells. But why would we bother going through all of this? And I think I alluded to a few of these. One is that much better mimic of what happens in vivo, so in the real human situation. And I'm a bit of a virus guy, so I like to study respiratory viruses. And there's good data to show that when you're looking at inflammatory responses, when you're looking at how the cells respond in terms of the pathology and the changes that happen after infection, the primary human air liquid interface model better replicates what you see in a human. The other important thing is we can measure all sorts of different functions that you can't measure in traditional cells. So because there's an air side and a liquid side, we can measure the barrier function. I showed you pictures of the cilia moving around, and that can be very relevant for different conditions. And we can also measure ion channels. So one thing I didn't mention is that these cells have sodium and potassium and chloride channels. And that's very important to keep your airway well hydrated. And so that can be measured as well. And I think a great thing to also understand is that we can mimic infections. So by that, I mean we can add virus to the airway and we can see what happens to the cells in a much more traditional way that these cells would face an infection. Um, and the final advantage that I think is super important is that because we're using human cells, when we see outcomes in the lab, we can then link them to humans, individual humans. So we will see different outcomes when we test cells from different people, and we can then go back and apply it to that person. Um, so this is big. I mentioned cystic fibrosis before, and this is something that we've done in the setting of cystic fibrosis, where we measure ion channel activity from cells from a particular individual, and show that specific drugs can correct that person's protein function, whereas they may not work in a different person. And then we start to think about how we can apply that in the clinical setting. So when you're thinking about human cells, there's different models that we've looked at. Explant lungs means these are lungs that come from lung transplant uh, individuals, and when their lungs are no longer required because they've got new donor lungs, the old lungs can be used for research. In the past, at least in the setting of cystic fibrosis and some other lung diseases, these have been considered a gold standard because there's a lot of cells that can be obtained, and a lot of the original protocols came from this model. The disadvantage is that they don't really have any relevance to the individual because that individual has now had a lung transplant. And so you're not going to suddenly change therapy based on this. Types of patients that will end up donating their lungs. And so if you're interested in a certain condition, this kind of limits you. Bronchial cells are the actual airway cells. So these are cells lower down in the lungs. The bronchi are just the breathing tubes. So they're interesting because they are the actual cells that we're interested in. They're the airway cells. They're definitely relevant to the specific patient because whatever results you find can be traced back to that person. And you could technically sample anybody you want. The downside is it takes some expertise to do a bronchoscopy. So you generally the person has some form of sedation, and certainly in children, they're usually a general anesthetic. And then there may be some mild risk to the patient with that anesthetic. So this isn't something that can be done routinely. And certainly from a research perspective, it's difficult to justify anesthetics for children. Induced pluripotent stem cells, I know you've heard a little, are a great model. You can have unlimited supply of these cells. They produce forever. They're relevant to the patient because they have the same genes, and they're pretty easy to sample in that there's good protocols to derive stem cells from blood samples. The downsides are they cost lots of money. You need lots of technical expertise to turn iPSCs into lung epithelium, and technically it's a source outside the lung. And then so with nasal cells, I've just put in why we like them. I've put also it's a source outside the lung because, nasal cells are different from airway cells, although we've done good studies to show that there's great correlation between nasal and bronchial cells. Um, and then I just put a plug-in for our CFIT program. If you ever want to get a sense of what we're able to do um, with our primary cells for a specific disease model, you can check out this resource, and it certainly helped us build this model. So, what can we measure in terms of in the lab that may be relevant? One of the first things is gene expression, and I know from an A&E perspective, people are very interested in genes. Initially, I'm showing you a figure here, which is basically single cell RNA-seq. So what that means is we're able to measure gene expression in each individual cell from our inserts. And for the scientists out there, I think part of this, what's great about this is we can do single cell on a single insert. And so we harvest the RNA, run it through the sequencer, and we can get stuff like this. What's important is that we see multiple cell types. So you can see everything. If you're an airway biologist, you'll see that we have different types of ciliated cells, basal cells, and even the rare ionocytes, which are only 1 in 1,000. And on this violin plot here, what I'm showing you is the colors match the different clusters or the different genes. And you can see here, or different cell types, I should say. And then here, you can see that we can segregate by gene. So we can see which cell types are expressing which genes. Um, Now, this is work I've done in collaboration with Christine Baer, and we haven't looked for RANDBP2, but this is something we can look at and see which airway cells actually have more or less gene expression. How does this change over time? How does this change with infection if we think it would? Or more importantly, what other genes in the pathway may change when we're lacking, a functional RANDBP2? So gene expression is an important outcome that is something to look at. The other thing is, and I mentioned to you that these cells are difficult to genetically modify, and they are, but we have been able to do this. So this is in collaboration with a guy named Jim Hugh, whose office was next door to mine. He has a vector or a virus that can introduce genes into cells, a helper-dependent adenoviral vector. So it's a virus that is the DNA from the actual virus is removed, and you can put in whatever DNA you want. And in this case, what Jim did is he made a virus that expresses a green fluorescent protein, so GFP. Um, and these are just three images here from the same cells. Bright field means just looking under the microscope and seeing the cells. GFP is obviously looking for the green signal. And DAPI is a blue stain that picks up the cell nuclei. So you can see that when we introduce this helper-dependent GFP, we can see lots of green cells. and they're all expressed here. So we can trick our cells into expressing certain genes. In this case, Jim also introduced CFTR. So remember I told you that's the protein that's broken in cystic fibrosis. And we did this in cells from individuals that have cystic fibrosis. So that's what these numbers and letters mean at the bottom here. Over here are cells from healthy control people, and these three bars represent cells from individuals with cystic fibrosis. These axes just represent CFTR function. So all I want you to see is that in the white bar where you have no vector, no gene therapy, there's basically no bar. It's very little function. When we add the CFTR vector, you can see that we have now function. The black bars are higher than the other bars. And this is the green fluorescent protein just as a control. So this is just to show that not only can we make the cells turn green, we can actually add uh, proteins by gene therapy to correct an abnormal function. And in this way, we can validate, gene therapy type vectors. The other thing we can do is we can measure lots of different proteins. And this is fairly easy to do with things called Western blots, which I'm not sure if you've seen any in the symposium so far. But we can also do very high throughput assays. So this is just a bar just to show you that, we talked about inflammation. We can use a Luminex assay and so we can add virus to the top of our cells. So that's what we did here. You can see the little RSV on the bottom here. So these are cells from a bunch of healthy control infants and also a bunch of children in red who were wheezing. These are kids that come to the emerge with wheezing. And then we grew their cells and we infected some of them with respiratory syncytial virus, a virus. And then we measured what happened in the media at the bottom of the cells. And so you can see that we can measure all sorts of different cytokines and characterize the inflammation. And for the most part, if you have the virus, you get more cytokines. But it's different between wheezy cells, individuals who wheeze and individuals who didn't wheeze. So we can start to look for patterns not only with infection, but also patterns that are different between individuals. There are new platforms where you can measure up to 3,000 cytokines. I'm not sure why we would ever do this, just to spend a lot of money, I guess. But if there was something that was interesting, you can really characterize like crazy these cells. In terms of viruses, so we've studied in our models adenovirus, coronavirus, including SARS-CoV-2, flu virus, and RSV. I wanted to show you a couple of things. Natalie, am I okay for time? I think I'm
Uh,
running a little over.
yeah, no, it's okay. Yeah, yeah.
Okay. I wanted to show you just how we can infect these cells. So this is a schematic of our inserts. So you can see the cells sitting here on the apical surface, and the media is at the bottom. And then this vial is supposed to represent virus. So what we usually do is we add the virus on the top, the same way that if you were a human running around, you would get introduced to the virus. It would come and hit you from the airway side. We let it sit for 2 hours, and then we remove the liquid and follow the infection for a few days. And this is just a course showing when we use an RSV, so a virus that we study routinely, but this one has been genetically changed so that when it infects cells, it turns the cells green. And so you can see after 24 hours, 48 hours, 72, and so forth, you slowly get our cells turning greener and greener, showing that this results in a productive infection. We can use this then and show that different things attenuate the infection. So this is work from Barbara in our lab who grew some macrophages. So these are immune cells that line the airway. And when she had the macrophages present, she could attenuate some infection, so less green. One of the things that the reason I'm showing you this is because we can also do co-culture. So Natalie, who was also in the lab, then ended up making this model a little more complicated, where instead of just adding the virus to the cells and having that as a simple model, she also had macrophages in the bottom. So in this way, these macrophages, these immune cells, can communicate with the epithelial cells and make the infection or modify the infection. So you can start to make this model a little more complicated than just epithelial cells. And Natalie did this, and she found that the type of macrophage made a difference. So macrophages aren't just simple one flavor, you can have what we call M1 macrophages, which are graded in fighting certain infections. And we can also have M2 macrophages, which tend to be a bit more allergic-y, for lack of a better word. But when we had more of the allergic-y macrophages, you can see that these macrophages allowed for much more virus to replicate compared to the more um, M1-ish macrophages. So that's another thing that we can start to do with our cells. I just wanted to show you a couple of quick things that I'm not sure if Alex already showed some of these data, but we've worked with Alex and Ava in his lab, and so this is I just wanted to show you a couple of things that we've been able to do, because it illustrates some of the stuff we can do with these cells. So I mentioned that the cells are difficult to genetically modify, but thanks to Alex and Ava, we were able to get from them um, some interfering RNA. So this is something that will genetically knock down expression of different genes. So Alex has one that can knock down RANBP2. And so here, where you see the little plus sign, these are cells from three different donors. And where the plus sign is, the cells were grown with this interfering RNA. And when you look down here at this Western blot, a Western blot, for those of you who aren't scientists, just a fancy way of saying I'm looking for protein using an antibody. So where you see a black band, there's protein. Where you don't see a black band, there's no protein. So here where we have the plus sign with the interfering RNA, there's no protein. So the first thing I want to tell you about this is that with Alex, we're able to modify our cells so that they don't express as much RANBP2 as in the normal situation. Now, I should say as an aside, kind of hidden in this data is the fact that RANBP2 is really essential for cell survival. And so when we're very good at doing this, we end up killing the cells, which isn't great. And so that's just one little caveat. But at least you can see in this model system that we're able to do that. The second thing I want to show you is that I told you that influenza A is an important virus, or influenza in general, I should say. So the other thing I wanted to just illustrate is we're able to infect our cells. And so these plus signs show that these are cells that are infected with influenza versus cells that aren't. And so then we have a nice model system to try and see what happens to these cells when they either have or don't have what we think is competent or enough RANBP2, and what happens to them when they're either infected or at baseline in terms of influenza. And Alex has a whole host of proteins that he's interested in, and this is something that we're working on, is trying to figure out how does this change the cell response. Um, one teaser, which is just very preliminary data, is just this looking at inflammation, and I think this is interleukin 6 on this axis, and so the higher it goes, the more interleukin 6 or inflammation we're seeing. Again, these are the cells that are either deficient in RANPP2 or competent, and with or without influenza. This is 8 days post-infection versus 24 hours post-infection. Um, and yeah, that should be HPI, not PHI. And then what you can see is that at least there's a sense that after infection, after influenza, you're definitely seeing IL-6 being produced, and maybe a little bit more when RANBP2 is knocked out. Um, the last thing I wanted to show on this slide is the fact that we've been able to obtain cells from individuals with ANE and then also study them in the context of influenza. These are data that need to be replicated, but you can see that in two out of three individuals with ANE, we're seeing a little bit more um, virus being shed in the apical surface. So TCID 50 is a lab way of measuring the amount of virus. So you can see that what we do with this is we actually wash the apical surface and measure the amount of virus. So there's a pretty consistent amount being produced daily here, but in these two individuals, we get a higher peak and then it kind of settles down. So these are some of the outcomes we can measure. I know I think I've been going a little bit over time here, but So in the end, I think part of the reason why I'm excited about working with Alex is that we can and Anne as well, because actually I should mention that we were able to link with our ANE subjects through Anye's clinical expertise and connections. Um, so why study them? Because we can explore epithelial mechanisms, we can start to validate some of the cell line observations that are made, and we can start to explore why some people behave differently than other people. And that's it. And thanks again for your patience. I apologize for all the technical stuff. I think next time I will definitely try to fly out to Paris, or not Paris, but France.
Thanks very much, Theo. That's super interesting. And it does fit in with the other talks that we've been having. So that fits in. I don't know if anyone's got any questions. So maybe I can start. Can you hear me? Can you hear me well?
Yeah, I can hear you. It's just a little bit quiet.
Okay. I was just wondering, with your data with the TCID 50, is that so do you know why that one individual did not come out? Have you got other data to suggest why that wasn't a sample that came out?
No, I don't have a good explanation, and I think it's very preliminary. It's something we will need to replicate to see if we will see that. What I can tell you is that when I mentioned the donor heterogeneity, that is something we see where especially with not just with influenza, but with the virus I'm more familiar with which is RSV, where we do see some inserts allow for much greater replication of virus compared to others. Um, and it may have something to do with non-RANBP2 mechanisms, so other innate immune genes. Our lab members often know which donors are, because we have a liquid nitrogen biobank with cells that are stored from different individuals, and sometimes they'll tell me, "Oh yeah, cell number blah 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 always gives us great results, and cell number blah 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 doesn't." And so there's definitely some genetic difference between these cells from different donors that allows for more or less virus. Um, but I'm not sure exactly what it is. I mean, when you look at the epithelium, there's so many different innate genes that are involved in immunity. Yeah.
Yeah, for sure. I'll just show you your audience. So.
Yeah. Hello, audience. I hope you're enjoying France. It's very rainy in Toronto today.
It's hot here. Um, yeah, you've got a question at the back, but maybe you should come here, Guillaume, because I don't think you can hear me.
Yeah. Hello. Just an open question because I pretty know the model of epithelial during my PhD, but
Sorry, I just can't hear you. I'm not sure why
ah,
it's so quiet.
Speak in the
okay, maybe
yeah.
near the computer. No, I'm just wondering because the epithelium it's a minimal model that it does not include immune cells, but you mentioned that you can introduce macrophage, which is for me pretty new in the field if I'm correct. But how do you suggest how do you manage to do this type of experiment in terms of personal medicines? Are you suggesting to grow separately epithelial cells from basal cells and then to differentiate them in vitro, and in parallel to introduce macrophage from the same patients? I don't know if it's already known in the field, or if you create another model with different type of macrophages. Just an open question about that, please.
Yeah, thank you. I think I heard the question, but just to be sure, you were asking about the co-culture model where we have the epithelium from an individual and then macrophages as well. So I think those are very tricky experiments, and it's exactly what you're saying in that there's different things you can modify. You can either have epithelium from the same person all the way through and then vary the macrophages. You can have different epithelium and then vary and keep the macrophage constant, or you can try and have macrophages and epithelium from the same person. We do have somebody in our institution, Martin Post, who has protocols for deriving alveolar-like macrophages from iPSCs. He's done this in mice, and he's done this in humans, and we've worked with him with the mouse system, and we're also collaborating with him in the human system. I think it does make for a fairly costly experiment, and so it would have to be something that you're really, it's kind of well thought out, and you know, you're thinking, why would I want to do this? And then the other variable that I think is important is our primary cells, like I told you, they were like babies almost. They like certain media, like the tissue culture environment, and the macrophages may prefer different media. And so you may start to see effects that are related to your experimental setup as opposed to, you know, what you think you're answering. And so it would be an experiment that would take a fair bit of thought if we were going to do iPSC-derived macrophages and nasal cells from the same individual, but certainly something that could be done. Yeah.
Thank you so much for the answer.
Okay, we have another question. I'm sorry, you have to come here. I don't know. I couldn't figure out the sound system.
Yeah,
It's surely
okay.
my fault. Yeah.
Great talk. Can you hear me?
Yeah, I can hear you.
Okay, that's great. Yes, so I was wondering, like, for that, I just go back to the IC50 experiment. I know it's quite pilot testing. So I have two questions about that. I think one is that, can you hear me?
Like he's having trouble.
Yeah, for some reason it's just very quiet, but
Okay. So I think the first question I have is that, you know, we know ANE has different genetic background. So for the three individuals that you look at, are they, you know, do they have the mutations or not? And then the second one is like, what, you know, is there any evidence or what made you think that, you know, these, the ANE1 patient will be more susceptible to virus as suggested by the experiment?
I think I heard the two questions. One was for the three individuals, did we know the variant? Was that the first question?
Yes.
Yeah. Yeah, we did. We do know the variant. I think for privacy things, I'm probably not going to say, but yeah, all three individuals have a RANBP2 variant um, and have variable clinical phenotypes. The second question was, is there a reason why they would have more virus? And I think that that's still something we're exploring. I think Alex may have some ideas as to some of the antiviral mechanisms and some of the proteins that may be involved downstream. But you know, I don't think the message is as much that there's an immune deficiency. I think it's more of a maybe a dysregulated response, maybe.
Yeah. Okay. All right.
Thank you.
Okay. So, Theo, thanks very much.
Great.
Thank you for your great presentation, and we're happy that you could come and talk to us about that. It's so interesting to hear what we can do now with medicine, even towards personalized medicine. So I don't know if you could hear that.
Okay. Thank you so much, guys. Enjoy the rest of the meeting.
Thanks, Theo. Bye.
Bye.